Good morning, everybody, on this beautiful Sunday, dear colleagues, dear friends and students. I'm happy to open the Symposium 6, devoted on toxins and ion channels, and we will have 4 speakers this morning. And I'm happy to open the floor for Dr. Felipe Zerni from
Você vai lá para a
Brazil.
Sociedade para a gente fazer os treinamentos lá até. Vai ter evento
He will
agora na.
give a talk on the understanding of reception elicited by diverse types of neurotoxins, PS5s, and their key contribution to stroke and drug-induced pain. We'll have a short introduction. Of course, many of you know Felipe already. He's a biologist with a master's and PhD in toxicology, with a clear emphasis on toxinology, performed at the Faculty of Pharmaceutical Sciences in Magneto, Houston. He conducted 3 postdoc fellowships, impressive. With a period in the Catholic University of Leuven, that sounds familiar, that's my home university in Belgium, and also in the Technical University of Denmark, ETU. Additionally, he was 2 years visiting professor at the Federal University of Roraima, and currently he's pursuing a postdoc position in the State University of Manaus in collaboration with Tropical Medicine Foundation. I hope I said this correctly. Felipe Zerni has expertise in toxinology, that's clear, but also immunology, electrophysiology, and federal bioprospecting. He has also dedicated himself especially to studying neglected diseases in indigenous communities and trade display, and all of this, of course, makes him an expert as an invited speaker today with the title I announced already. So the floor is yours, Felipe.
Thank you for your presentation, Titiga. It's an honor for me. Good morning, everyone. I'm glad someone woke up early this Sunday. I'm super glad for that, for coming here to listen to me. So I will talk about the disease non-accepted response induced by different classes of tissue-cellular neurotoxins. The important role of TS5 in the venom-induced non-reception. I will try not to have problems with this, but you see. First, I will do a briefly introduction about scorpions. So, the scorpions are the quite ancient group of our planet, with the fossil records dating back about 400 million years. So, in this article, we can see this evolutionary doesn't have too much significant anatomic changes. You can see the fossil. And the one explanation for this successful survival is the fact that they adapted to different types of ecosystems, such as forests and also deserts. The explanation for this is because the scorpion has a lot of resistance to survive a long period without food and water. They able to survive until 30 days without water and also 400 days without food. So we can find the scorpions around the world, with exception in the south and north poles. We call it the scorpion-free ecosystems. The reason is because it's too cold to allow this kind of species to survive here. Currently, there are more than 200 species described in the literature. They are distributed across 12 families and more than 200 genera. And there are around 34 species is with medical importance. And most of them is inside the Butites family. Highlights for the genera Iônicos, Butus, Centroides, Mesobutus, Hemiscorpus, Leiurus, Parabutus, and Titus, the fox always study. The genera Titus in Brazil. In Brazil, the genera Titus has 22 species. Some of them is very famous for us, toxicologists, like Titus baiensis, Mentuentus, Obscurus, Silvestris, Stigmurus, and the Titus cerulatus. For those who doesn't know, Titus cerulatus is this scorpion, is a small one, is around 7 centimeters when it was an adult, and the color is a mix yellow and brown. And the reason of this name is because it has a serratio in the metasoma near the skin. You can see in the picture here. These species are present in 15 of 26 Brazilian states, including here, Brasilia. And the one explanation for this widespread species is because they usually do a parthenogenesis is a one-sexual reproduction as well the fertilization. And also, it's so common to find in the nature just females. Additionally, the tropical climates of Brazil helps the proliferation. We are warm country and also have a lot of rain seasons. And the species in special is very, very adapted to urban areas. The reason of this is because they don't need to find a partner to do a reproduction. And also in the cities, we produce a lot of trash, and the trash attract a lot of insects, especially the cockroaches. That one, it's the perfect diet for this kind of scorpions. Indeed, just in 2022, 171,000 scorpion stinks are reported. That one represents 62.8% of the accidents caused by venomous animals. This data is from government Sinam. However, the medical importance of Titcerulato is not only related to the high number, but also because the venom it's have a high toxicity. I bring this article is not so new. You saw yesterday from Professor Arantes. I like the title because is a good definition about this venom. Because Titcerulato venom is, in fact, a lethal cocktail. In fact, just one sting in the human being can be damaged in the all biological system. And the reason this happened is because in the venom have a lot of compounds that one, such as like peptides, enzymes like hyaluronidase, metalloproteases, serinoprotease, and the stars of your study, the neurotoxins. We have the sodium toxins and also the potassium toxins. More than 20 toxins were isolated from Titcerulatos, and we have more than approximately 41 identified by transcriptomics, like you saw yesterday with Professor Arantes. And we know the toxins already described. It's just the top, the iceberg. We have more compounds, one node. So about the envenomation, it can be classified into mild, moderate, and severe. Mild manifestations are severe pain, paresthesia, sweating, nauseous. Moderate manifestations are tach and bradycardia, hypotension and hypertension, vomiting, diarrhea. And severe manifestations are cardiac failure, pulmonary edema, and death. So, independent of in the venomous severity, intense pain is always observed in the Titcerulato stings. Actually, the reports from the victims, they said when receive a sting in the foot, they can feel the pain on the leg more than 24 hours. And they induced mainly by venom-derived neurotoxin, able to modulate the sodium channels and the potassium channels. Titcerulato venom is composed by neurotoxins that bind specifically with to voltage sodium channels, such as a TS1, 2, 3, 4, and 5, already tested and published. As well, a neurotoxins target voltage gate potassium channels, such as TS6, 7, 8, 9, 15, TS fragment 1, and TS fragment 2. Our group was pioneer to perform electrophysiology of several Titcerulato toxins, such as TS1, TS2, TS4, 5, 6, and 7 in the same article, TS8, 15, and TS90 fragment 1 and 2. Although many Titcerulato toxins activate ion channels associated with pain, like Navy 1.3, 1.7, 1.8, 1.9, and KV 4.2. And few studies have focused on the noroception response, with only 2 have been conducted with isolated TS compounds. One of them is from our group, TS8, and another one from a different group. To understand the noroception induction to Titcerulato venom, we tested the noroception response of isolated toxins from the main classes of neurotoxins. So for alpha sodium tox, alpha and beta sodium tox, we tested TS5 and 1. For alpha sodium potassium toxin, we tested TS6. And for beta potassium tox, we tested TS8 because we already have data, we already published. And we test a new toxin, TS fragment 2,
when I say new tox, because I discovered this, I identify these toxins in my PhD. As expected, the toxins acting on Navy channels, TS1 and TS5, induced significant nociceptive behavior after 10 minutes of injection. Like you can see here, representing the purple and the blue arrow. The purple is TS5 and the blue is TS1. And TS8 Xvii toxins that modulate Xvii 4.2 channel also induced a significant and delayed nociceptive behavior, as you can see here, represented with the red arrow. Interesting, the whole venom of titocerulates showed the significant and the less pain behavior with a similar connection to TS5, as you can see here, and in this graphic also. So to perform the nociception testing, we measure the pulse leaking time after injecting the toxins. In this case, I bring the video that one the mice who are we are injecting TS5, and it starts to leaking when it feel the pain. You can see it's frenetic leaking because it have a lot of pain, and also we put like this mirror around the jail, I don't know the correct name, to check it, because sometimes the mice start to walk around. You need to check to measure the time. It's not so exciting, but when you finish everything, it's super good to see in the graphic. And use a different also, when I use a different quantities of TS5, the nociceptive behavior demonstrates to be a dose dependent. Like you can see here in the graphical, this is a control, is a venom, and this is a TS5. You can see the both, when you increase the amount, they both increase the line. And this intriguing result made us the question, is TS5 toxins the main responsible for the pain caused by titocerulate stink? So, to test this hypothesis, next we produce an anti-TS5 polyclonal neutralized antibody. For this, we did with 3 immunized animals. Only one of them, the name mouse 2, was able to produce a detectable titles of anti-TS5 antibodies, right? Like you can see here in this area. The serum of M2 mouse was used to isolate anti-TS5 IgGs, and mouse IgGs were purified properly, as can be observed in the SDS page gel, you can see here. And after purification, the IgGs showed they still recognize TS5 through the ELISA. You can see in this red square. To neutralize the TS5 from the whole venom, we consider the percentage of TS5 is recovered from the titocerulates venom. This is around 2%. We did this with software help to do this. And based on that, we calculate anti-TS5 IgGs 2 times 2 equimolar of TS5, and which was pre-incubate 1 hour and at 37 degrees with 2 equimolar IgGs anti-TS5. We know that IgGs are bivalent, and the ratio of binding site and the TS5 was 4:1. So they have the site, you have a 4, and it is a toxin. And a significant decrease in the nociception response was observed when compared to the animal that received just the venom without pre-incubates with the anti-TS5 antibody. We can see here in the graphic. Regarding the group that received isolated TS5 toxin pre-incubate with the anti-TS antibody, the nociception response was completely abolished. You can see for our good surprise, is completely abolished. And from the for the indirect ELISA assay, what demonstrated that scorpion antivenom was able to recognize titocerulates venom and also the toxin TS5. You can see here the arrows represent that, and indicated that specific Brazilian scorpions antivenom maybe decrease the pain in the venomation victims. To the in silico analysis with TS5 toxins, suggest 4 predictable epitopes that will code responsible that antibody binding. So, in our conclusion, this study, our explored the noroception behavior induced by different classes of scorpions, neurotoxins, alpha and beta sodium toxins and potassium toxins, and demonstrated the important role of TS5 in the Titius cerulatus venom sting. This work is already published in the PLOS Neglected Tropical Disease Journal. Published is not so recently, in January of this year. And we'll talk about a little bit what's the next step. So we are evaluating venom compound and the nororeception induced by different scorpions from Amazonia. So we do this with Abroteas amazonicus and Titius metuendus. Both can be found in the Amazonas state. And also, we do this with Rhopalurus crassicauda and Titius silvestris. Both can be found in the Roraima states. And for our surprise, we found two new species. We identify 2 new species of scorpions. That one is not described in the literature. Both we found inside the forest in the Roraima states. It's not so easy to go there to pick up these scorpions. And the data is not published yet. We work in the article. Maybe this year or begins the next year, we will publish this. I would like to thank the organizing committee to invite me today. A special thank for the Professor Puca from eTox Lab from NESP. Special thank also for Professor Arantes from the LTA Lab from USP. Professor Tiago from USP, Professor Monteiro from Medicina Tropical de Manaus, Fundação da Medicina Tropical de Manaus, for another co-authors, and the FAPEAM, Fundação de Amparo à Pesquisa do Estado do Amazonas, for the scholarship to support me to do the research. And thanks. If you have some question, I'm glad to answer.
Thank you, Felipe, for this excellent and state-of-the-art lecture. We have time for questions, perhaps first from the audience and then also online, if there are colleagues interested to ask questions.
If you have some questions later, I'm happy to answer in the Congress.
Thanks so much. Okay, then we can continue with the second speaker of this morning. I'm happy to introduce Emílio Beraldo Neto from Brazil. And he will give a talk dealing with the prospecting and achieving comprehensive neuroproteomic study of alpha-KTX12 insights into potassium channel blockade. And to introduce him, let's say, more decently and further, Felipe helps me here. So Emílio is a biomedical graduate from the Methodist University of Sao Paulo. He completed his master's degree in the pharmacology lab and his PhD in biochemistry, both at the Instituto Butantan at Science Technology program. He is currently carrying out a postdoctoral position in the Department of Biophysics in the Sao Paulo School of Medicine at Unifesp, where he develops a project with the objective of early diagnosis of leukemias using mass spectrometry analysis. Recently, hired as a young researcher at the biochemistry laboratory at Butantan, he develops his lines of research in prospecting new bioactive molecules from poisons and venoms, acting mainly in the purification, biochemical identification, and pharmacological characterization. And of course, with this expertise, he's excellently suited to give us his talk of this morning. The floor is yours.
Hello. Good morning. I want to thank the organizing committee for inviting me to this lecture. Thank you, Dr. Mariana. Thank you, Professor Jan. Today, I'm going to talk a little about the beginning of my research line. As Professor Jan said, I I am a young researcher at Butantan, and I would just let the anxious get out from the body now. And well, my line of research is prospection of bioactive molecules from Venus and secretions. And the main find of my objective is to find alternatives for neurodegenerative disease. And prospection, you already have a lot of lectures how the prospection works. So what we do is try a lot of molecules, and to hit one molecule, one simple form, one simple experiment, and then we develop the study on this molecule. So I would like to thank my students. They helped me a lot. Without them, I would not be able to do this research. And part of the results that I'm going to present is from Laís Campello PhD project. So just to give you some introduction here about the neurodegenerative disease, it's an increased disease in number of patients. It's affect most people over the age of 65 years old. And as we know, with medicine, the population is getting older and older over time. And here we have a graph of every continent of the population over 65 years old. I'm not getting the pathophysiological of the disease. I am not there yet. Okay, to experiment on specific disease. I will explain how I am conducting my research line. So first, we have a simple and comprehensive model where we prospect the venom, the fractions of the venom. When we select one fraction or one molecule, in this case, when I send molecule, I send focus on the peptides and the small organic molecules. When we select this particular molecule, we go to more specific models, yet not the neurodegenerative disease. It's more some kind of injuries or something like that. We try to, when we see some potential here, we go to unveil the mechanisms of this molecule. So here we are not trying to find a target, a receptor of this peptide, but to see the response of the tissue that the peptide is working on. The presentation here will stop at the number 4 here, yet not in the specific neurodegenerative model. Okay. So I have separated the presentation in these 3 parts to be more didactic: the prospect, the find, and the development. The first, well, we are working with this presentation will be based on the Titus baiensis venom. Is one of medical importance scorpions in Brazil. And this is his reversal phase chromatogram. I have separated this chromatogram in 2 different ways. And the first is this 83 peaks that I individually made the proteomics of these peaks. And the second way is the fractions that's separated by these red lines to test the biological assays. Okay. Well, from the proteomics, we have identified several peptides that act on sodium channels, more than 15 isoforms. And for potassium channels, we have found at least 8 or 9 isoforms. We have found a lot of enzymes too, like hyaluronidase, metallopeptidases. But I'm not going to focus on all these peptides here. It's to show these works that are published. The prospection part we did with SAH lineage. It's a neuroblastoma cell. So first we prospect in the fractions of the venom. We don't like to start with all venom because as you all know, the venom it's very rich, and together with all the molecules there, they have a synergic effect. So we like to fractionate to don't have this cytotoxic effect. And here we have found, in this model, we were just trying to look for something to continue the work. Here is the first prospection. So what we have found is the fraction tree have increase in cell viability. So we already knew what's in the fraction tree. I had the proteomics. So we did the isolated peaks of the Vano, and have found that the fraction 3.4 was the molecule that had the effect. So the fraction 3.4 is an isoform of KTX12 that blocks Xvii 1.3 and 0.2. We have made the LCMS analysis to see the purification. Here you can see the Maltov analysis, the sequence. This is I called isoform because it's not the exact same sequence from the original one described. It has at least 2 different residues alteration in 600 Dalton less, so it lost some residues. And this analysis here is the LCMS to see the purification. Here we can see another peak, but it's the same peptide we have checked is without one residue that it lost, so that the peptide lost. So with this information, we go further in the analysis. We have testing another model. Then the first model, so it was the viability test. So the peptide increased the viability in that model. Here's the PC12 lineage itself. Here we have made a better analysis. We have made a curve of dosage, the concentration of the peptide. We have seen that in 5 nanograms by ml, the peptide decreases active metabolity, but increases cell integrity. And we have checked also ROS, but it's not increased ROS, so that's a good thing. We have made a temporal analysis to see not the peptide prevents the lesion, but if can recover a lesion from a cell. So first we cause the injury here by hydrogen peroxide, and then 4 hours later we introduce the peptide in the cell culture. Is the same lineage here, the PC12 cell, and what we found was not the best result that we want. But here you can see 2 things. In ROS, we do not have an increase in ROS in time. Integrity, you can see these 2 lines here at the bottom. The top one is the peptide with hydrogen, is the peptide, and the bottom one is just the hydrogen peroxide group. So besides this close, it still have a statistical significance. So and this is made with 5 nanograms. Maybe if we increase this peptide here, we could improve this result. This is still an ongoing work from Lai's project. So you had there are already 2 different lineages, the SCH lineage that have an increase in viability cell, the PC12 that have an increase in integrity cell, not when compared to a hydrogen peroxide group. But we want to test more this peptide, so we go to animal analysis. The chosen model was to test this particular peptide in hippocampus of the animals. So for that, we have performed stereotax surgery and intrahippocampial injection of the peptide. And we have performed 2 analyses with this, the histology and the neuroproteomics analysis. And for neuroproteomics, we have surgically removed the hippocampus, the cortex. The cortex is all the lobes, external lobes, and the cerebellum. And for proteomics, we have 3 control groups: the negative control, that's saline, and a positive, that LPS. For histology, I don't have the LPS group for ethical committees things. So for histological analysis, I have two different analyses here. This first one is the qualitative analysis. So as you can see here, it's just the negative control group and the experimental group. So the red circle here is the pycnosis cells, as we call, is the damaged cells, and we can find a lot more damaged cells in the control group than in the alpha catechis group. And so why is this happening? Why our control is more damaged than our experimental group? This is a basal damage caused by the surgery. And what we think is that the alpha catechis may be being neuroprotective in this analysis. But it's still just a how to say, qualitative analysis. The quantitative analysis, as we count the health cells, not the damaged one, you have seen the increase in relation to control. Remember, the control suffers little damage as the experimental one. This analysis is made with a manual count. We have count 3 slices of each animal. We have 6 animals here. So all the regions that have count, that see 1, 3, and 4 regions of the hippocampus are increased. The ipsilateral side is the side of the injection. The contralateral side is the other side of the hippocampus. And after that, we have performed the neuroproteomic analysis. The neuroproteomics generates a lot of information, but we are trying to see what the peptide is doing in this tissue, right? So what I'm going to present here today is just the 6 hours hippocampus analysis. We have performed label-free analysis. This is already a filter result. I have removed all the inflammatory proteins because the LPS group. So we have excluded a lot of proteins here. These are the work data. And from these proteins, we have performed the functional protein interaction in string. As you can see, this does not answer a lot of things. It's very complex, but help to understand the interaction between the proteins. So I have to facilitate the understanding of what's going on in the brain, I have separated the proteins in several groups. These groups are mitochondrial membrane proteins, snare complex. This is glutamate pathway proteins. This, according to the literature, is neuroprotective proteins. And this last one is the enzymes of energetic pathway proteins. Okay. And this last one here is just a better visualization of the increased proteins of the label-free. So to summarize the data, for my understanding, for me, I have made this figure here. So where you can see that the peptide is blockading the channels. This is called, all the information I'm gonna talk about today is from proteins that I have found in the neuroprotein canals, okay? So we have found proteins that are trying to organize the ions inside the cell, in the inner part of the cell, measure the hydrogen ions, okay? So you think that maybe causing a depolarization in the mitochondria, that we have found the 2 pathways, energetic pathways in of the mitochondria diminished, the ATP synthase and the acid secrete pathway. But we have found the glucose cycle increased. We have not performed an ATP dosage here to see what's going on, but this according to the literature, is very common in neurons, because the glucose cycle is the preferred pathway to neuroplasticity of the axons. So it's a good sign for the peptide. So we have found also the increase of protein translation. These are gonna go after. So I have found the neurotrophic factors, neurostructure, proteins, axonal growth, axonal growth. I not measure this, but it's proteins that related to axonal growth. Okay, enolases 43 proteins. That is always related with the glucose cycle increase. And what I have found is the decrease of malate aspartate shuttle. This pathway is related to glutamate production and recycle. The glutamate is the major neurotransmitter of the brain, but in increased levels, it can cause damage. That's called excitotoxicity. And it's very common to see in this kind of methodology that we perform that the toxins increase the glutamate and cause this kind of damage. Okay. So here we have not made the dosage of glutamate yet, but is in our list. And you have the neurotrophic factors and proteins related to neuronal plasticity. So just to conclude the presentation, we have in 3 different models, we have seen increase the cell viability, cell integrity, increase the number of parameter cells, and a suggestion of maybe what's going on in the brain that we are evaluating. And I just want to thank you, everyone in my lab, for this work. It's not just mine. This is my group of students that are here today. This is the other collaborators, the financial agents, and the other universities that helped me with the analysis. Thank you.
Okay, thank you, Emilio, for this excellent talk. Again, we have time for questions. Yes, hello.
Thank you, Emilio. Good data. I have one question about the damage in the hippocampus was a peripheral injection, okay? Was direct
directing the hippocampus. We have
Directing
performed a surgery to do the surgery.
Stereotaxy.
Yes.
Okay. I have a question. Are you thinking about a plan to test the behavior? Because you could damage the whole limbic system, not only the hippocampus. If you put it over there, it can spread and go to the amygdaloid system and everything. And another question is probably could be a good model to test the memory behavior in the animals. Okay, this kind of cells can be involved in behavior of memory and other behaviors. Okay.
Yeah. Yes.
Congratulations. Could be a good model in the future.
Yeah. Well, I'd like to test all of it, but I still don't have enough students or time to do so. About the behavior. When we performed the first surgery injection that I have shown, I didn't do any specific analysis, but the animals after half hour of the surgery, of the injection, they were normal. They are eating, drinking. I don't perform any specific tests. What?
It takes time.
Yeah, it takes time to analyze everything. Yes. Yeah. Yeah. But after the surgery, what was the second question? Sorry, what's the second? A segunda pergunta?
Thank you. It's the only question about if you are thinking of test this. Okay.
Yeah. Okay.
Thank you.
Thank you. Thank you for the question.
Emilio, nice talk. Thank you. I have a question about the protocol. Did you run the proteomics from the 6 mice individually?
No, I have pulled the samples and run in technique replicates. I have performed the same proteomic in the pooled sample 3 times to gather
the same sample.
The same sample.
Okay. Thank you.
Okay, Emilio, I also have a question.
Okay.
So if the general aim is prospection of using peptides for neurodegenerative diseases, so can you comment on our concern we should have in terms of the knowledge of these peptides being able to cross the blood-brain barrier and reach the CNS at all? I mean, can we do these days predictions from the sequence that you say, these peptides has a kind of cell-penetrating additional added value, or isn't that a concern in your studies?
Well, I still not evaluate that, but what I know about the literature of these peptides, they do not cross this barrier. I still not there yet, but it's something that I will concern in the future. Like the model that I'm gonna choose for all that I have presented here is to choose a specific model to test for neurodegenerative disease. So I don't get there yet, and I still don't know how to deliver the peptide yet. For experimental models, I am delivering by a simple surgery, but for the future, I still don't know.
Okay, thanks.
Thank you.
Do we still have other questions from the audience here, physically present? No? Do we have questions online? I don't know how we get informed if there are. No? No questions online, thanks. Okay, let's join me in thanking Emilio once again for this nice lecture. So without further ado, we can continue with the third presentation of this morning, delivered by Dr. Manuel Aguilar from Mexico. And even when he's not physically present, so he will do a remote and online contribution, for which we are very happy. He will talk about a short M conotoxin from the venom of a worm hunter snail, Conus archon, that inhibits human neuronal nicotinic acetylcholine receptors. And to introduce Dr. Manuel Aguilar Ramirez further, he was born in Xalapa in Mexico and studied as a pharmaceutical chemist and biologist in his hometown. Then in Mexico City, he did his bachelor's thesis and completed a master's and a PhD studies in biochemistry at the Faculty of Chemistry at the National Autonomous University of Mexico. In March '98, he moved to Querétaro, in Mexico, of course, to work in the Marine Neuropharmacology Laboratory of the Institute of Neurobiology of the National Autonomous University of Mexico, founded in 1996 by Mr. Heimer de la Cotera. And he pioneered the toxinological studies of turret snails worldwide and of cone snails and cnidarians in Mexico. Then a bit more recent, in 2013, he was promoted to associate professor, and he assumed full responsibility for the laboratory when Dr. Heimer became advisory professor. And in view of the proven utility as molecular probes and models for drug design of several peptide toxins from cone snails, known commonly as conotoxins, inhabiting the Indo-Pacific region, his long-term research and interest and plans proposed the study of the diversity of toxins from cones and turret snails species as a whole. And from this background, of course, we are very glad also on this conference that he accepted to contribute his, let's say, latest finding on the title I mentioned to. So, Manuel, I hope you can hear me from Mexico, but the floor is yours, and we are ready here to listen to your talk.
Thank you very much, Jan. Nice to see you, and I want to thank the organizing and scientific committees of the Congress for inviting me to present our results. Can you hear me? Excuse me, Jan. Can you hear me?
In that, when it started, you did correct, but we turn off the chair. You need to put in the presentation mode. Could you put this? Could you do this, Manuel?
I'm trying. Can you hear me?
Yes, we can hear you, but we can see you. You can't see you.
Okay.
Yeah, now it's perfect.
But can you see the
Presentation,
presentation?
you need to click on the presentation mode in the right corner in the bottom. Because we see yours. Yes, now it's perfect.
Okay. Okay, thank you very much again to the organizing and scientific committees for inviting me, and all of you for helping me to broadcast the presentation. This is a recent work we conducted in our laboratory in Querétaro. As an introduction, most of you know snails, but I will just say that not all the marine snails are venomous. Some of them are, and they use their venoms for hunting prey and self-defense. And among these venomous snails, there are the cone snails that acquire their name from the shape of the shell. I will refer to the species simply by conus, which is a general name for the genus. Recently, this genus has been divided in several subgenuses, such as Profundiconus, Californiconus, Conasprella, and Conus. These snails prey on worms, mollusks, and fishes, depending on the species of the cone snail. And the venoms contain small molecules, but the main components are peptides, which are called conopeptides or conotoxins. Some distinctions have been made depending on the content of cysteine residues. These conotoxins or conopeptides affect muscle and nervous system of their prey, or also of predators. And the main molecular targets are voltage and ligand-gated ion channels, but they are also other targets such as neurotransmitter transporters, G-protein coupled receptors, insulin receptor, and enzymes. We decided to start studying this species in a particular way because most species of conus have been shown to produce inhibitors of nicotinic receptors, both competitive, such as the alpha conotoxins, or non-competitive, such as the psi conotoxins. In general, the alpha conotoxins affect muscular and neuronal subtypes of the nicotinic receptor, whereas the psi conotoxins affect the muscle subtypes. These receptors are important because they participate in the release of neurotransmitters and therefore are involved in diverse pathologies such as Alzheimer's and Parkinson's diseases, schizophrenia, epilepsy, inflammation, pain, and other conditions. Therefore, conotoxins that affect these receptors are interesting ligands as probes to study the structure and function of these receptors, but also can be leads for designing therapeutic agents to treat these conditions. The species we are working now is Conusarcon, is a worm hunter that can be found in waters from the west coast of Mexico to Central America, up to Panama. And this species has not been studied from the toxinological point of view. Here we have a nice picture taken by a colleague of the University of Sinaloa from the shell of this species. Conusarcon belongs to the subgenus Stephanoconus. I show here a close of a phylogenetic tree published by Pulander et al. Some years ago, in which the position of Arcon in this tree is highlighted by the red arrow. And it is well known that several species in this subgenus produce nicotinic acetylcholine receptor inhibitors. In this slide, we can see several toxins from Conus regius, Conus sonatus, Conus imperialis, and Conus bruneus. And in a very simple way, I try to show what types of nicotinic receptor these toxins affect. Some affect neuronal and muscle subtypes, whereas others affect mainly neuronal. Thus, knowing this, our objective was to identify and purify conotoxins from Conusarcon that affect nicotinic receptors, and then to purify some of them to characterize them from the chemical and electrophysiological points of view, and to perform initial characterization of the three-dimensional structures. While we prepare a crude extract from the venom duct by homogenization and centrifugation, we use reverse phase HPLC to fractionate the extract, employing a C18 column. And we screened the fractions by electrophysiology assays using Xenopus oocytes expressing several human subtypes of the nicotinic receptors. We focus on the most abundant fractions and the most active, as I'll show you soon. And then we characterize the molecule by mass spectrometry and automated Tillman sequencing. And then we try to assess the selectivity of this toxin by testing it on other subtypes. The initial subtype was the alpha-7 subtype, but we we'll see that it affects other subtypes in a lower degree. We built a 3D structure model, and we used simultaneity search to find a proper template for building this model that was refined and evaluated by this program modeler for building the model and refined to refine the initial model. Okay, these are the results. This is a chromatogram of the venom dog extract. We highlighted here the apparently most abundant fractions, which were tested on the alpha 7 subtype. Here we have a fraction 11, that affects the oocyte and apparently inhibits significantly the acetylcholine-induced response of this subtype. However, we couldn't continue studying it because it damaged the membrane, and the oocyte dies very quickly. Other fractions, such as fraction 38, has no significant effect on this subtype, and these other fractions, 55 and 57, don't have a significant degree of inhibition. Our system has let's say, an experimental error of about 10%. So to be sure of continue working with a really active compound, we did not continue studying these fractions. However, fraction 27 was active, and then we decided to purify it to characterize it in more detail. Here is the several steps of purification on the C18 column. And then we determined the molecular mass of the peptide and the amino acid sequence. We used a transcriptome which has been prepared from this species to determine whether the C-terminus is free or amidated, because the Edman degradation doesn't allow us to know this. And sometimes the mass spectrometry is not enough to know this in a absolutely sure way, depending on the equipment. So when we compare the sequence of the purified peptide with a partial transcript from the transcriptome, we see that the precursor ends with an arginine residue, which is removed by proteases, and therefore the C-terminus cannot be amidated in the toxin. It has a free C-terminus. The experimental mass agrees very well with the theoretical mass, considering also 3 disulfide bridges among the 6 cysteine residues that the toxin contains. And this is the effect of the purified peptide on the alpha-7 human receptor. It has a interesting effect because the inhibition is almost irreversible. You can see a small degree of recovery, but I think there are 37 minutes here, and the increase in response to acetylcholine is only about 7%. These traces show the effect of the toxin on other subtypes, both human alpha-3 beta-2 and alpha-7 beta-2. The effect is not significant in the case of alpha-3 beta-2. In the case of alpha-7 beta-2 is not really spectacular. And for now we are not studying more the effect on this subtype. However, despite the limitations in the amount of natural peptide, we were able to build a concentration response curve on the alpha-7 subtype. And in this graph, we compare this dose-response relationship with the effect of the toxin on the other subtypes at a concentration of 36 macromolar here for the 3 toxins, but we have more data points for the alpha-7 subtype. And the calculated IC50 is about 46 micromolar. Here we have the 95% confidence interval. Okay, well, we wanted to know more about this toxin, which is very, very small. Fortunately, the simultaneity search revealed a very high sequence similarity with a toxin whose 3D structure was recently determined by NMR, and the sequences are almost identical. Only one residue is different in 15 residues at this position, serine instead of a threonine residue. Both are polar residues. So the model built by Modeler is very similar. We can see to the right the comparison between the model and the template. And well, I present here some details of the evaluation of the model. I think is acceptable. Most of the residues are in allowed regions and most favored regions, and only one residue in disallowed regions. Actually, apparently the model is better than the template. And well, at the bottom, I present two views of the model of this toxin. Because of the effect on nicotinic receptors, we compared our toxin with toxins with the same cysteine framework, number 3 in Roman letters, such as the Psi, P3E, 50F, and Alpha M3J, Psi PRR3E. All of them affect nicotinic receptors, but here we can see that the similarity between our toxin and any of them is very low. On purpose, I didn't highlight the cysteine residues to point out to the non-cysteine residues, which are similar in gray or identical in black background to our toxin. The most similar toxin is in this case is alpha conotoxin M3J from Conus magus. You can see that 3 non-cysteine residues are identical with our toxins, but unfortunately the structure of this rather small toxin is not has not been determined. So in this slide, I compared our toxin in panel A with 2 Psi conotoxins, P3E and P3F, that affect the muscle subtype of the nicotinic receptors. Whereas our toxin affect neuronal subtypes, especially the alpha-7. From this comparison, which is made at the same scale, we can see that the structures are very different, and therefore it's not surprising that this toxin from Conus arcon affect neuronal subtypes in contrast to the Psi conotoxins that affect the muscle subtypes. Here is a table comparing several details of these toxins, the number of residues, the connectivity of the disulfide bridges, the number of residues between subsequent cysteine residues, and a branch subtype or group. Several terms have been used to classify these M conotoxins or mini M conotoxins. And here we have the type of inhibition. In the other toxins, the effect is clearly reversible, whereas in our case we could consider it irreversible or very slowly reversible. And we can see here also difference in the receptors that have been used to test the toxins. Some are from torpedoes, from mice, from zebrafish, human, etc. Due to the structure and the size of our toxin, we compared it with a small framework 3 toxin from Conus literatus, also a vermivorous species from the Indo-Pacific that affects sodium channels. Is a iota conotoxin, and we can see that only the cysteine residues are shared by these toxins. So although we didn't have enough material to test our toxins on sodium channels, it seems very unlikely that our toxin can affect sodium channels. At least in the iota conotoxins activate sodium channels. But there are small toxins with the same system framework that affect, that inhibit, not activate, but inhibit the sodium channels, the muconotoxins from piscivorous species. Here we have the alignment, and again, the sequences are very different to our sequence. Only one residue is identical if we don't consider the cysteine residues. So our toxins doesn't seem to be an inhibitor or blocker of sodium channels. So the conclusion of this study is that toxin Arch3A is the first conotoxin characterized from this species. And at the same time is the first M-conotoxin from a vermivorous species shown to affect nicotinic receptors. Also, this peptide is the first mini M2 conotoxin that has been shown to affect nicotinic receptors. It should be mentioned that the molecule that was used as a template to build the 3D model, its function or target has not been identified. This toxin inhibits the human alpha-7 subtype more than the alpha-7 beta-2 subtype. Its plausible 3D structure is compatible with this effect on neuronal receptors. We saw the comparison with toxins that affect the muscle subtype, the psychonotoxins. And for now, we can see that it is unlikely that this toxin affect sodium channels. For the future, we plan to synthesize this toxin in order to have enough material to perform other experiments, such as testing it on other subtypes. I put here some subtypes, alpha-4 beta-2, alpha-7 beta-10, for example, to see how much selective is this toxin. For now, we only know that it differentiates or distinguishes between alpha-7 and alpha-7 beta-2, but there are other, and it doesn't affect the alpha-3 beta-2 subtype. We also want to determine whether this toxin is a competitive or a non-competitive inhibitor of nicotinic receptors. And of course, we would like to determine experimentally the structure by NMR to have a better model of its structure. Which project was funded by the university, the National Autonomous University of Mexico, and a grant program from the same university. I thank you very much for your attention, and I show you two views of the Institute of Neurobiology, where most of these experiments were conducted. We are celebrating 30 years of the foundation of this institute. Thank you very much.
Thank you very much, Manuel, indeed, for this very interesting talk. Please stay with us because we may have some questions here from the audience, first of all. Do you have questions for Dr. Manuel? I look around. If not, if you allow me then to ask a more general question from my side. We all know that conus species have a very elaborated post-translational modification machinery. Can we today make predictions, let's say, in a particular clade like the Stephanoconus, or on the other hand, worm or fish hunting conus? Can we make predictions in terms of the degree of post-translational modifications that we may expect, or is that still unknown? And is it always an ad hoc observation?
Yes, I'm sorry. I did understand the last sentence, the question.
So when we try to identify novel conotoxins, can we already make predictions in the degree of post-translational modifications to expect if you consider, let's say, worm or fish hunting cone snails, or if you focus on the Stephanoconus, or is it still too early?
Well, some post-translational modifications can be relatively, can be predicted, but others are not so easily or confidently predicted. In the case of our toxins, well, the molecular mass and the chromatograms of the Hitman sequencing clearly show that this toxin doesn't contain any post-translational modification apart of the disulfide bridges.
Okay, thank you. Other questions from the audience or maybe online? No, if not, please join me in thanking Dr. Manuel once again for this very nice talk.
Thank you very much.
Bye-bye. Bye.
Bye, Jen.
Good morning, all. It's a pleasure and an honor to introduce Dr. Jan Tittiger. Jan Tittiger is a full professor in toxinology and head of Lab of Toxicology and Food Chemistry at the Catholic University Leuven, Belgium. He holds a bachelor's and master's degree in pharmaceutical sciences and continued his career by obtaining a PhD in physiology at the Medical School of the KU Leuven. From 1999 to 1992, he has studied at the Harvard Medical School, Boston, USA, for a postdoctoral training. He holds the chair of toxicology with tuition and research in different areas of toxicology in the faculties of medicine, pharmaceutical, and agriculture science. He is also director of the division Biopharmaceutical Sciences of Leuven Research and Development, a bio-incubator for his research in drug discovery. Jan Tittiger has published more than 150 peer-reviewed articles and reviews in international scientific journals, and has received several prizes, among which the prize of the Research Council, KU Leuven, the most prestigious prize of young scientists at KU Leuven, and the international prize Dr. E. de Croix of the Flemish Marine Institute for his researches in the area of cnidaria intoxications and drug discovery, starting from marine systems and organisms. Jan Tittiger is also leading a Forsync toxicology lab in Belgium at the request of the Ministry of Justice. He has been elected president of the European section of the International Society on Toxinology 2004-2011. And he will present the talk, histamine receptors, ex vivo functional studies enabling the discovery of hits from venomous animals. Jan, thanks a lot for being here.
Thank you. Thank you very much, Dr. Beth, for this very kind introduction. Dear colleagues, let me first thank the organizers, Professora Mariana, and the committee, but also the scientific committee, for giving me the chance and opportunity to present here this ongoing work in the laboratory. And apart from the scientific, let's say, contributions, it's always really a true pleasure, not only an honor, but also a pleasure coming to Brazil, even when it's a far, let's say, voyage and distance for a short time. It's always with so much pleasure to see familiar faces here with whom we have been successfully collaborating in the past years. And many of you have of course become friends, and this is a nice sign of let's say element of science without borders, where in these difficult times in the world where people suffer and have problems with neighbors, we as scientists can give an example that science, true science across nations and the borders is really something to pursue and to cherish. So it is a true pleasure being here. And like Dr. Beth already introduced, I will focus on something novel that in the course of what I do in the lab for almost 30 years now has become more and more of interest. And these are receptors, at the same time sometimes also ion channels, so they definitely fall in the category of this session. And I will focus today on the histamine receptors, where we, over the last years, have established an ex vivo functional study enabling the discovery of hits from venomous animals. And the work I will present is also, let's say, if I can, it is being part of a PhD student of Andrea Seldeslags, who is not here, but she will defend her thesis next year. And then also in support and collaboration with Steve Peigneur, who is physically present here. And many of you know Steve because he's a long-time collaborator and postdoc in my lab, for which I'm very grateful. So the three of us, in fact, have contributed to this work. Let me first, for a couple of minutes, guide you a bit to a short pharmacology lecture. What are histamine receptors? Why are they interesting, especially in the field of toxinology? What can we still expect in terms of drug discovery and toxins? Well, histamine receptors, the first thing you have to know, they of course are G-protein coupled receptors. And in our genome, we have four, let's say, locuses, histamine receptor 1, 2, 4. So as a pictorial, you have to imagine we talk about membrane-bound protein, of course. It's a typical feature of a GPCR, so when 7 transmembrane helices. And upon binding of a ligand like histamine, they crosstalk with a very interesting cytocellular mini-protein, which also, you know, the G protein, which has been described for many years in textbook as trimeric. So you have the G protein consisting of an alpha, beta, and gamma subunit. And when GPCRs are activated by their respective ligand, in this case histamine, a kind of conversion of this trimeric protein occurs, and GTP that is present in the cytosol is being hydrolyzed, and this is like the signal to also dissociate or let dissociate this trimeric state of the G protein. And then in the literature, mainly the effect of the alpha subunit has been described. On the one hand, I will explain, but also the effect of the beta-gamma, let's say, counterpart as well. And what do we know? So the alpha subunit, what has been described for histamine receptors pharmacology, is when you focus first on the histamine 1 receptor, people say it activates a kind of G-alpha-Q pathway. And in this pathway, it's important that you will see a phospholipase C activity with the conversion of PIP2, phosphoinositol phosphate 2, into inositol phosphate 3, IP3, also the diacetyl glycerol. And this ultimately leads in the cell to a calcium concentration increase. Then the histamine 2 receptor, there people believe when the alpha is released and active, it couples to a G-alpha S type of pathway, where now the aniline cyclase system is becoming activated. And as a consequence, you have an increase of cyclic AMP, and we all know this is kind of a secondary messenger, and it will activate a kind of phosphorylation of the type phosphorylation kinase A. And this is also important because phosphorylation of many targets, like for instance some sodium channels, is important for their function. And then you have the histamine 3 and 4 types. They are a bit less well studied. They couple what the literature says to a G-alpha IO type of G protein, and literature predicts that they now inhibit the analyte cyclase pathway, leading to the opposite of the H2 receptors, meaning so you have a decrease of the cyclic AMP. So that has been described for, let's say, the consequences upon alpha activation. But also in our study, you will see we focus not only on the alpha, but primarily on the beta-gamma consequence in this pathway. And it's known that beta-gamma is doing something very interesting. They will, let's say, target some other membrane-bound proteins, which are ion channels. And one of the important channels are what we call G protein coupled inward rectifying potassium. So that's what we call jerk channels, G protein inwardly recting potassium. And you have many types of these. They have been cloned, and we study primarily the type 1 and type 2 isoform. And when beta-gamma, let's say, is bound to these jerk channels, you see an opening of this potassium channel, and depending on the driving force, depending on whether you have a lot of potassium concentration outside or inside, you can expect outward current netto or inward current. That depends. So that's common knowledge in the literature. And the last thing that is important also for our, let's say, platform that we developed are cytosolar proteins from the family of RGS. And this means regulators of G protein signaling. RGS. And we also have cloned these available in the lab. You have 4 isoforms or orthologs, 1 to 4. And what they do is they, let's say, if you apply an ex vivo system like they mimic as much as possible the in vivo situation. So in terms of this GTP-GDP conversion and then re-establishing of the GTP, let's say, load, they take care that the kinetics are very much alike, like in an in vivo cell system. Okay, what about their expression? Because we can think about toxinologists. Okay, this is pharmacology. What is it? Why is it useful to focus also in the field of our beloved field of toxinology? Well, if we see where are they expressed, let me first start with the H1 type. There, primarily, we know it's very important in the smooth muscle cells. H1 receptors in the smooth muscle cells, but not uniquely. You can find them also in the central nervous system, in the pulmonary system, in the heart. So they are quite abundantly expressed. For the H2 receptors, we definitely have to focus on the gastric mucosa. So the stomach mucosa, but also there you can find expression of H2 histamine receptors in different type of blood cells, in the central nervous system, in the heart as well. Then the H3 receptor, there it's less known, but people say it's important in the central nervous system primarily, apart from being present in epithelial cells in the gut, in, of course, the lungs, again, in the heart. And then the H4 receptor, it's more, let's say, a kind of orphan type receptors. We know very, very little. People say it's in the kidney, in the gut, but some blood type cells, but we have no clear picture what the relevance is. But of course, when we talk about histamine, we think about mast cells that we all have and are potentially becoming activated upon intoxication with all the sometimes nasty consequences of the histamine release. So from this aspect, any interference from a toxinology point of view with either one of these 4 receptors or all of them will have, of course, a clear effect on mast cells and then the clinical symptoms derived thereof. So what about the medication? Is this still novel? Is this uncharted territory? Have we medication worldwide? Yes, we do. And I have to focus on the fact, and you probably know this, they're always antihistaminics. So we have examples of successful medication worldwide on the market, but they always counteract, let's say, the histamine effect or are antagonists of the receptors. Let me first show well-known antagonists for the H1 receptor, worldwide used, diphenhydramine, then the second generation loratadine, further generations hydroxyzine, cetirizine. I guess people know this against, let's say, allergic reactions, and where then the first generation had the side effects of sedation and sometimes depression. And this has then, as we go along with the development of the newer anti-H1 receptor antagonists, been, let's say, solved to have less side effects. But worldwide, they're used immensively. What about H2? Well, think about the mucosa and the stomach. There again, oops, we have the press twice. Okay, thanks. We have the ranitidine and the cimetidine, also widely used for people with burning pain in the stomach and problems with the proton pump. Because pharmacologically speaking, these molecules are proton pump inhibitors. Then the H3 receptors, a bit less known, but there is one on the market. It's called pitolisant, and pitolisant or pitolisant is known to treat narcolepsy in humans, in adults. You see the example of the guy here. And yeah, there are also side effects, but there is only one example on the market. But then, surprisingly, for the H4 receptor, we have none. We have no antagonists, and we have no clear picture what is H4 receptors in terms of the mast cell activation and the release of histamine is doing. So what is then the link between histamine and the small pharmacological introduction I gave you and the topic of our beloved science, toxinology, and venomous animals? Well, I take as example today a caterpillar. A caterpillar where you all will agree with me, if you get a physical contact, mainly via the skin, it won't be your best day because you suffer from inflammation, you suffer, oops, go back. No, sorry. The bottom, sorry. Okay, so if you suffer from inflammation, pain, and skin rashes and itch, it's not your best day. And definitely, if we can have a medication and where also toxinology inspires us, it would be an added value. And this brings me to, indeed, the animal of today. It's a caterpillar and species known as Automeris zaruma. It belongs to the family of the Saturniidae. It's a moth that at an adult stage becomes like a butterfly, but we call it a moth. And at the stage of its caterpillar, it's truly covered with spines, as you can see here, very, very carefully looking spines, and they serve as a storage room for a venomous cocktail. People know that these spines, of course, can be called stinging spines, so be aware, don't touch them. They are everywhere on the caterpillar, let's say on the animal, dorsal, subdorsal, and they can really be very impressive to 0.5 centimeter in length. Scanning electron microscopy examples are known, and then it's very clear-cut that you can consider all these spines as a kind of cocktail of mini needles. Mini needles, hollow structures that upon physical contact break, even, of course, after penetration of mainly your skin. And we were then very much wondering, what is then the venom cocktail within these spines? Can we discover new toxins that eventually are related with histamine release? You have to know that, in general, there are only supportive treatments for Automeris zaruma caterpillar envenomations. People say, you have to remove the setae, you have to remove the spines with a sticky tape. You have to avoid scratching. You can apply topically corticosteroids. Sure, that will help. But you know, corticosteroids, they do have side effects, in particular when you apply and use them for more than 2 or 3 weeks or months, we will know. And the use of antihistaminic drugs currently on the market, there is a true scarce efficacy. So there is place and room for improvement. So what did we do? We were interested in this Automeris zaruma venom at the caterpillar stage. And the way we approached is threefold. So of course, we have mass spectrometry in the lab. We have a very nice QTOF MS where we can do proteomics. Then, and this is the talk of today, we, let's say, over many, many years have specialized in learning about the bioactivity of the venom, in developing platforms, so to discover what are the key targets in the pathway. And the ultimate goal, of course, is to go from target to therapy. It would be nice to dream of a kind of lotion or cream or whatever medication to bring on the market to help people suffering from such an encounter with these caterpillars. So the experimental setup and the main talk or part of my talk today is these ex vivo functional studies. And even when this technique has been around for decades already, I still want to very briefly introduce you because the potential of this technique is still enormous, and I will give examples. So we have established over, let's say now 30 years in the lab, a true extensive cDNA library, meaning we have the genes, clones, that encode for many receptors and channels, I will show in a minute. We can make, with the classical molecular biology technique, so-called runoff transcripts from T7, SP6 RNA promoters. And then when we have this collection of RNA, here these red, let's say, marks, we can inject them in nano concentrations in the oocyte. And the oocyte is then given from, of course, female animals from the Xenopus levi system. And after a couple of days, these oocytes are very, very, let's say, cooperative. They do the translation. And remarkably still for me today is that they know how to process this particular translated proteins into the membrane. And then when you truly have overexpression, we apply and rely on the electrophysiology technique, so the classical 2-electrode voltage clamp. And this 2-electrode voltage clamp can be executed with microelectrodes. That's the most, let's say, common and simple approach. Then you do a whole cell measurement, so you measure all the activity of channels and receptors over this oocyte. But you can also apply in the voltage clamp mode, the so-called patch clamp. Then you do not use microelectrodes, but then patch electrodes, where you then simply want to observe, let's say, the activity of one such green protein shown here. The advantage of this system is that it's very robust, it's open, and thanks to the development of an high efficient, let's say, plasmid expression vector, you can do truly overexpression. And this is important if you do molecular pharmacology, functional pharmacology, that you can say, what we study here is truly this or that ion channel, and then the effect of the toxin is this or that. So to give you an idea about the cDNA library, because when I was starting like 30 years ago in the lab, and when I came back from the United States from my postdoc, we only, or I only could do voltage-gated potassium channels, the shaker type KV1. That was it. So the human ortholog, or the rat, I should say, the rat ortholog, and then the insect from Drosophila. And that was it. But nowadays, we have in our library not only the shaker type, but also shaker, Schau, and Schall, so KV2, 3, 4. We have KV7, interesting, and perhaps also as much interesting, the EAG and the HERG channels, so KV10 and KV11. And these channels are important also for, let's say, preclinical toxicological studies. So not only for drug design, but also to test a particular metabolite, compound, medication, drug, toxin, to show that there is, for instance, no interference with a cardiac potassium channel, and that can give you a safety prediction that it will not introduce like long QT prolongation and the long QT problems associated. Also, we have recently published with a Brazilian group that some of these potassium channels are modulated in particular types of cancer. So when they are upregulated in cancer cells and you can, let's say, block them with a particular toxin, it's also a nice diagnostic tool. So we have, in the field of voltage-gated potassium channels, you see many isoforms that we can express. Alike in the family of the voltage-gated sodium channels, you have to know that we as humans, we have in our human genome 9 isoforms, and they are called NAV11 to 19. And in the previous talks by Emilio, we have already had a glimpse of that. The importance here of these NAV channels is that they are quite often organ-specific, like in the skeletal muscle, you have 1.4, in the heart, you have 1.5. And if you're interested in pain, which is a big issue, of course, in the science, then some of these sodium channels are true pain sensors, like 1.8, 1.7, 1.9. So when you overexpress, you can do really a kind of niche pharmacology there. We also have voltage-gated calcium channels nowadays, and we focus on the T-type calcium channels. They are called calcium V3, 3.1, 3.2, 3.3, and they're important in pacemaking activity. So when our heart and, let's say, natural rhythm is there, the basis of life, in fact, we can also study potential toxins that can have a bradycardic or tachycardic effect via these clones. And alongside with that, we also have since several years the so-called cyclic nucleotide-gated or true pacemaker channels, hyperpolarization cationic nucleotide-gated channels 1, 2, 4. We can express them also in the oocytes. And curiously, after many, many years of efforts with some PhD students, so far we have not found any animal, venomous animal, poisonous animals, or toxin acting on these pacemaker channels. So it would be very nice if we still can find a selective molecule there. We have then also inwardly rectifying potassium channels not coupled to G proteins, and they are important for establishing membrane potentials. So all our cells have a particular membrane potential, neuronal cells more negative than, for instance, smooth muscle cells, minus 90 millivolt inside versus outside. For neuronal cells, smooth muscle cells, it's more maybe minus 50, minus 60. And these guys really regulate this, let's say, balance. Then for the topic of today, we have then the true G protein coupled potassium channels. They look very much similar to these IRIC channels, but I call them HIRIC channels, 1, 2, and 4. We can also express 2-pore background potassium channels like TBAC, and they have been implicated with more recent literature by other colleagues in the field of cancer, again, in the field of pain. And these channels, so the inwardly rectifying, the 2-pore background, they're much smaller. They don't have like this voltage sensing apparatus like sodium and calcium channels have. They're very much smaller, but again, in terms of pharmacological discovery and toxin field, still much to explore. And then we have also many accessory subunits that you need, like the beta subunits to express successfully sodium channels, like a min-K, which is a minimal single transmembrane segment that we co-express, for instance, with this KV10 or KV11. And then these famous regulators of G protein signal I introduced you already. So that's a whole bunch, but more and more, we become also in our lab interested in the ligand-gated channels and receptors that you can just as well express functionally and study with this oocyte system. So for the moment, we have all the GABA-A receptor subunits. We have many nicotinic acetylcholine receptor subunits, so we can express a neuronal type, like in the talk of Dr. Manuel, the alpha-7, but we can also mix and match and express a muscle type. We have many TRP channels, also important. For instance, the TRPV1 is very famous, the vanilloid or the capsaicin receptor, also in terms of thermoregulation and pain sensation. We have also the ENaC channels, important in the kidney. These are the amiloride-sensitive epithelial sodium channels that we can express. We have also several purinergic receptors, and these are, let's say, not well known as ion channels as such. We call them receptors, but when a ligand appears, they open and they are, let's say, physically an ionotropic channel. Then you have the true G protein coupled receptors, so the histamine receptors, which are the topic of today. Then you have also a mass-related G protein coupled receptor that is very, very novel, and I will show you some unpublished. Oops. Sorry. I will show you some example also today. For many years, we have the opiate receptors, mu kappa delta. They express very nicely in oocytes, important, of course, in pain. We have the cannabinoid receptors, and here I refer for those of you who attended yesterday to the lecture of Steve Peigneur. So we are also interested in novel venoms and toxins acting on CB1 and CB2, because worldwide, you will agree with me that there is an interest in medical or medicinal cannabis, and that many countries also try to regulate and legalize, let's say, the recreational use of cannabis. So we believe that also in the field of toxinology, we hope that we still can discover some unique ligands acting either on CB1 or CB2 and learn from that. Then we have also the bradykinin receptors, important in terms of itch and pain, and then some serotonin receptors. And then to summarize the list, we also have invertebrated or invertebrate targets like some potassium and sodium channels like the glutamate receptor. And for all of this, us interested in pesticide treatment and management, when we rely on, let's say, toxins produced by spiders and scorpions and other creatures, well, of course, the platform that we offer with the oocytes to be able to functionally study the invertebrate targets is also definitely an asset. And if that's not all, we are also now capable of expressing some bacterial targets, like the NAGBAC. This was the very, very first sodium channel crystallized and from which the structure was published, so it's a bacterial. And many, many toxins that we applied on this sodium channel from bacteria, it's completely resistant. So do not think that even when the sequence of a bacterial sodium channel is very much alike ours, the pharmacology can be really, truly different. And then the last one, of course, with the pandemic that we all know, please be aware that also our system is able to check functionally viral targets. So we can very nicely express so-called viroporins. If you think about the SARS-CoV pandemic, SARS-CoV-E virus produces in these spikes, particular spike proteins. One of those is the E protein, and it's a channel. So what you can do these days is simply order, let's say, the synthetic gene that is known from the genome. You put it in an expression vector, you make RNA, and then see when the oocytes can express it, and it works. So there also, the platform we propose opens a huge diversity, as you can say, from the cDNA library here. And I don't want to advertise perhaps too much, but in case of some people here in the audience or listening to the talk are interested in checking one of these targets that we propose, feel free to contact me. I'm happy to listen. Okay, coming back to the topic of today, identify the targets. So you have to do a validation, of course, and we did a quite extensive validation. So on the left, you see non-injected oocytes, yes? And we do electrophysiology, so we measure a current. So in many traces, you will see a kind of scale bar. Vertically is the amplitude in microampere, and horizontally is a time scale. So you take a non-injected cell, and you apply a decent concentration of histamine, or you apply a concentration of the venom of this caterpillar, and you put it in high potassium, which is just let's say solution you use to measure. And the first thing you hope to see is no effect. Okay. No effect, so a flat line. So you know the oocyte as such doesn't react. And then you say, I'll take, for instance, the H4 receptor, and I will co-inject this with these famous Hurk channels and also RGS4. So I refer to my introduction. So imagine you have now this nice overexpression of this histamine 4 receptor. You have also co-expressed HIRC1 and 2, and you have co-expressed RGS4. You rely on the G protein of the oocyte. So the G protein itself, you don't inject, it's physiologically present. And when you do this on the right, what is then the effect you hope to see? Much different from the negative signal here. Now you see the following. When you apply in blue here a high potassium solution, so you change the driving force for potassium to have a better signal. You see an inward current. When you apply histamine, you see a very, very nice activation, which is controllable because if you wash out the red or the histamine, you go back. And if you go back to the physiological solution in green, you go back to the starting level. So this is a very nice, let's say, validation experiment of knowing, okay, do I really see H4 receptors activation and can I control this, yes or no? But what about then the G protein coupled potassium channels by themselves? You can say, well, maybe they determine everything of the reaction. So then, of course, we do ex vivo functional studies where we only express HIRC1 and 2. And here you see 1 micromolar histamine has no effect. Also the related, let's say, brother or sister clone IRK1, no effect. And further, you all know from pharmacology, I hope, this famous pertussis toxin that is well known to block the dissociation of the trimeric state of the G protein. When you co-apply pertussis toxin and you do apply histamine, you hope and you would expect not to see anything, which is there. So that's a kind of pharmacological validation that the system works. And then, of course, we started with the venom, the Automerus zaruma venom, and we first checked the H4 receptor. So on the left, you will always see the control. So you go from a physiological, what we call a 96 buffer, to a high potassium, just again to increase your signal. You see now a potassium channel influx. You apply histamine, you wash it out, and you can go back. So that's the control. And much to our delight, when we applied the caterpillar's venom, we saw a very, very similar activation. So something in the venom is there that can activate the H4 receptor. And as a control for the histamine, we determined a very, very, let's say, sensitive, high affinity EC50, 13 nanomolar for the H4 receptor, which is very sensitive. So then we continued to the H3 receptor. H3, I can be brief, qualitatively the similar effect, so histamine and then the venom, and here the IC50 or the EC50 was 24 nanomolar. We continued with the H2 receptor, and much to our surprise now, again, the similar reaction. And why do I say to our surprise? Because the literature doesn't predict that H2 receptors couple to these GRK channels. It's not shown. And so we browsed the literature, and it seems that activation of several GPCRs is in fact more promiscuous than what people have been told in general in the literature. So do not think about a unique pathway being activated only by GIO or GS or GQ. Sometimes they can follow many pathways together. And this is an example. So also histamine 2 receptors, they can couple to a GIO system with then the potassium channels involved. However, the histamine 1 receptor, that is different. Histamine 1 receptors do not couple to these potassium channels, and then you can wonder how can we study this in oocytes? Well, we can. For the histamine 1 receptor, it is known that they will activate an increase in calcium, so release, but also influx. And in oocytes, it's known if you have an increase of calcium, you will activate mainly chloride channels that we didn't inject that are endogenously present. So you would expect when the venom is active via H1 receptors, that there is a calcium-activated chloride current. And that is exactly what we saw. So on the left, in control with histamine, so the black trace is in control, you apply histamine, and you see an incredible increase, here the outward current, which is in fact an inward-going chloride anion current. And the venom was also being able to introduce that via H1 receptor activation. What about selectivity? And I hope this is not too small, and I will not dwell on that too long. The selectivity is also, of course, an issue, because if nature has designed the venom of the Automerus aruma caterpillar venom in the way to uniquely activate histamine receptors, then we should be able, again, with the oocyte system, to test it. So here on the left upper part, A and B, these are traces of nicotinic receptors, just like in the case of the previous talk of Dr. Manuel from Mexico. So here I can summarize, no effect was seen, neither on a muscle type or neuronal type of acetylcholine receptor expressed in the oocytes. You can apply as much as venom, no difference with as compared to the normal acetylcholine activation. What about ASIC channels? I didn't talk about that. We can also express these. These are acid-sensing ion channels, so they are important in ischemic conditions. They react as a pain sensor too. And since an encounter with caterpillar venom on your skin also causes pain, we checked it, no effect. So we can exclude ASIC activity. Alike, we can exclude TRPV1 activity as a pain mediator. We can also exclude the bradykinin that I introduced to. So, and we also did, of course, always the proper control. So the blue is with bradykinin, and you see again here a calcium-activated chloride. In red, with the venom, nothing happens. And the same with the capsaicin receptor. So we apply capsaicin, and you see a very nice inward current. You apply the venom, and nothing happens. The same here on the left with the cannabinoid receptors, where also Steve talked to you about today. This is an example of the CB2 receptor, where we, in control, apply it and activate it with WIN. It's a classical agonist. When you, on the other hand, apply it with the venom, no effect. However, there is this orphan receptor that also took our attention. It's called the mass-related G-protein coupled receptor X2, uniquely expressed in our genome. And this was, let's say, activatable by a classical agonist, a small molecule 48/80 that is known in the literature. So we bought it and it's working. And to our delight, also the venom is acting on this orphan G-protein receptor. So allow me to give you one more slide on this. The mass-related G-protein coupled receptor X2 is uniquely expressed in humans. It is abundantly expressed in mast cells that we have. And it is assumed to be responsible for inflammatory pain. And when you activate this receptor and let it activate also this giric potassium channels, you talk about non-IgE mediated pseudo-allergic reactions. Because the mast cells activation, of course, there is also a clear allergic pathway, or the EGE related pathway. Here it's a non-EGE mediated pseudo-allergic reaction because you target really the receptor. So the molecule available in the literature, this 48/80 is doing it, but also the venom. And which venom components are hidden in the venomous cocktail. So we did an Q2 MS analysis, and apart from the proteomic analysis, we focus today here on the small molecules. And indeed, in Automeris zaruma, we were able not only to identify, but also to quantify histamine in the venom. There is 1 micromolar of histamine in 0.2 microgram per microliter venom extract in this species. So we can clearly understand why it activates. However, and this is still the big question mark I have, when we talk about this mass-related G protein coupled receptor, this so-called orphan GPCR, when we did a control, we applied histamine, nothing happens. And yet the venom is capable of activating this enormously. What is then the molecule behind in the venom that activates this MRGPC6 receptor? We still don't know. And it's, in my opinion, very important to elucidate this, because you have to look at histamine or mast cells as bombs filled with histamine. And yet the venom doesn't seem to be able, with histamine, to open this bomb and release this major, let's say, histamine release in the body with all the consequences. So there should be something else, and we are still exploring it. So, but for me, a very fascinating question mark to resolve. How can we treat it? And I try to conclude. So I told you in the beginning, there is a scarce efficacy on the market of the current antihistaminic drugs. From our platform, one, let's say, quantitative result that we didn't expect was the affinities. Look, H1 and H2 receptors for histamine, you talk about micromolar affinity, whereas 3 and 4, very much less explored in pharmaceutical drug discovery, you talk about nanomolars. So when you have circulating histamine in the body, primarily these H3 and H4 receptors will be, let's say, targeted first. That's clear. And this we can also prove by some molecules that are on the market but haven't reached, let's say, a phase 2 clinical application or commercially available. There is one compound known as a Johnson & Johnson compound, 777 and then 120. And indeed, when you apply histamine as control on the H4 receptor, you apply the venom, as I've shown before, then you can nicely show that when you apply this J&J molecule, and then thereafter histamine or the venom, nothing happens. And this molecule is rather lipophilic, so even when we wash it out, the antagonistic effect remains. So there is potential to have small molecules as antihistaminics or anti-H4 receptors, but it still should be explored. So this brings me to the mast-related G protein coupled receptor. Think about a mast cell. Think about the non-IgE dependent pathway. So then you have to think about all these small blue receptors. The mast cell itself is really a bomb full of histamine, and it waits until a trigger, either via immunological pathway or, let's say, the receptor-based pathway, to release its content. And be aware that some companies are already very active in this field, and sometimes they are not aware from our research on from a toxinology point of view. And I give one example. In California, in San Diego, there is this small biotech company, Escient Pharmaceuticals, and they really focus on this mass-related G protein coupled receptors. And as you can see on their homepage, they nicely summarize the importance of this receptor in terms of noxious stimuli that there may be from the airways, from the skin, from the GI tract. They can activate many pathways in an immune adaptive system or from the CNS, because also from the CNS, they are interested in the role of histamine receptors, in particular this orphan receptor, in even let's say, pathologies of dementia. They have already one molecule almost on the market. It's called EP262, where they claim it's the redefinement of treatment of mast cells, cell-mediated disorders. So you see there is from the pharmaceutical industry definitely an interest here. So I come to my conclusions, dear friends, dear colleagues. We have established and validated an ex vivo screening platform enabling the discovery of hits and leads that modulate histamine receptors and this mass-related G protein coupled receptor X2 in humans. However, neither H4 antagonists nor MRGPC2 antagonists are in clinical use today, which definitely should be considered and encouraged. We believe also that more research is also needed to explore the role of our MRGPCs in mast cells, especially with venom intoxications. We have no clear clue, in particular with this Automeris caterpillar. What is the molecule? What is the peptide behind that is capable to activate so efficiently this receptor? We don't know. So the knowledge will be useful for the treatment of other dermatitis and inflammation-related diseases, we believe, apart from caterpillar envenomations. And with this, I want to end. The venom itself from this nice caterpillar was given by Dietrich Meebs, that you all know. He's of course now emeritus professor, but so active for many, many years in the IST Society. And so I'm grateful here to here on the left, Andrea Seldeslags, that's a PhD student. And then on the right here, Steve, that is also physically present for helping with this project. So this is the end. I thank you for your attention and I remain available for your questions you may have. Thank you.
A very nice talk, Jan. Thank you very much. We are open for questions. Paulo?
As Elizabeth said, very nice, fantastic talk. And in the final of your presentation, you put some more consideration about the neurons terminal. If they put the venom from this caterpillar in the skin, one of the first thing the venom does is stimulate the terminal axon. And this increases the amount of P substance, the peptide, the P substance to release histamine from the mastocytes. That's you didn't test the P substance in your oocytes with the express main receptors. I missed this test because could be combined with the histamine and the venom from the caterpillar. What do you think about this?
Excellent question. And I agree with you. This is only the start. A very fundamental. Should work. It's only the start. Definitely, it more. That was also part of my conclusion. It deserves more elaborate studies, including the diverse pathways that are implicated in the pathologies of pain and itching and inflammation. And substance P, of course, is there also a crucial parameter. But honestly, we cannot answer for the moment because it is still, let's say, at its childhood's let's say, stage.
Manuela?
Thanks, Ian, for the presentation. I learned a lot about these new receptors. It's just a question, is this the first venom that connecting these both receptors, histamine and the MERG? Or we have another venoms? Do you suspect that other venoms could also act in this receptor?
Well, to the best of our knowledge, in terms of molecular pharmacological proof, yes. We have in the lab also other caterpillar venoms, and initially we saw also effects. However, some of them also activate the G proteins by themselves, which is also an interesting, let's say, result. And therefore, you have to do the proper validation that to know that the venom here, let's say, has no action on, let's say, this G protein coupled on the potassium channels, I mean, and that the effect can be explained solely via activation from the beginning on the receptor. But to answer, yes, I'm convinced there will be certainly other examples of venomous creatures, maybe not only caterpillars, but different ones that think about jellyfish, for instance, that also have a particular, similar symptoms. So to say, this all needs still to be explored. Yes.
Perfect.
We have question, Maria Helena?
Thank you, Jen. You ever present very nice talks. I would like to know if you have an idea of the chemical nature of this substance. It is proteic or non-proteic?
I can only speculate for the moment because it's ongoing, but I would not be surprised it's a small molecule. And the reason why I believe in this, but this is hypothetical, is the prototypical ligand that we can buy from Sigma, etc., that is being used as activator for this mass-related orphan protein receptor, this 48/80. It is a polymer of an phenylethylamine methoxy substitute. So it looks very much like a drug and like a neurotransmitter. I'm also, a forensic toxicologist, so my chemistry knowledge about recreational amphetamines and phenylethylamines goes back, and I was very much surprised that the polymer of this molecule is being used as prototype agonist for this mass-related G protein. So why not assuming that also in the venom of the caterpillar, we do not talk about a peptide or bigger molecule, but a kind of phenylethylamine structure, which would be no surprise, because if you look at histamine, you talk about almost molecules in the same family. But again, this is hypothetical, and I hope maybe on the next meeting we can elaborate on that.
Thank you. And do you think it's possible that this substance is present in other caterpillars? Probably no.
Yes, but we cannot generalize. We are also busy in the lab with a project with the processionary moth. I don't know in Brazil whether you also have problems with these caterpillars that form a processionary, let's say, chain. But definitely in many countries in Western Europe, in particular seasons, people have a lot of problems in terms of encounters and intoxication, also domestic animals, serious problems. It's a processionary moth. Is it known in Brazil? I don't know. But to make a long story short, there we have no indication that there is a major interaction with histamine receptors. There it's more with the TRP channels. So I'm still open to learn many discoveries that even within the family of the Saturniidae and the moths and the caterpillars, we may see different bioactive molecules activating several pathways.
Thank you, and congratulations.
Any other question? Jan, thank you so much. It's impressive, the library that you have of ion channels and receptors. I have a small question. Do you know how this venom is prepared from caterpillars? It's milked or it's the whole body?
Well, there too, there is not a clear-cut generalized ID. Can we call caterpillars venomous animals, or are they poisonous? Do they have a venom gland? How is the venom gland, let's say, connected with the setae or the spines? There is still some controversy, and I compare this with ribbon worms. Ribbon worms, they also have wonderful bioactive peptides, but can we talk about a true venom gland? Maybe yes, but it depends on how you define then anatomically a kind of layer and subdermal cells that indeed are capable of producing these molecules and then being in contact with the spines. I've been in contact, of course, with Dietrich Mebs, who is more specialist on that and who also took the nice EM photographs. And he believes it's really a venom that is in contact with the setae. And he also believes that if we talk about hemolymph also in insects, for instance, that there is like a physical connection sometimes with the hemolymph and, let's say, the venom gland of all these animals. But again, this is a bit controversial for some scientists as well.
Okay, thank you. Anyone? So thank you so much, Jan, for this nice talk.
I'd like to invite you to go outside the building for the group photo now, okay? And then we have the coffee break. Function of Dynamics Laboratory at the Instituto de Medicina de Valencia. And since 2011, Dr. Calvetti is president of the European section of the International Society on Toxinology. You were? Ah, okay. It doesn't say. Okay, not updated anything. Okay, sorry for that. Well, then he is editorial board member of Toxicon, several journals like Toxicon, Journal of Venom Research, Toxin Reviews, Biochemie, and editor-in-chief of the Journal of Proteomics. He belongs to the board of directors of the Global Snake Initiative, an internationally active non-profit organization registered in Australia and led by snakebite experts who are dedicated to improving access to good quality, robustly tested, safe, effective antivenoms in the world's poorest communities. He is a member of the WHO Technical and Scientific Advisory Group on the development of target product profiles for snake antivenom products, and is in the list of experts for implementation of the WHO strategic plan for control and prevention of snakebite envenoming. He co-authored more than 550 scientific publications in peer-reviewed journals and monographic books, which received over 23,000 citations. His current research focuses on evolutionary and translational proteomics of snake venoms, having developed mass spectrometry-based tools for exploring the evolution, composition, interaction with venoms, and biotechnological applications of venoms and toxins. Well, I now have the pleasure to invite Professor Calvetti for his talk. Please, Dr. Calvetti.
Thank you for the kind introduction. Questions? If I can have this working. Okay, so this is the title of my talk. Almost the people that you see here in the list are the current members of the lab. So we are a small lab, but try to be active, you will see. So the topic is venom and interaction with antivenoms. So let me start first with a conceptual slides and also to present the tools, the biotechnological tools, the proteomic-based tools that we're using to do what we do so that you understand what we are doing and why we are doing in this manner. We all know that venom has evolved in every ecosystem where there is competition for resources. There are venomous. There are species that use venom for getting their lunch. So, in these venoms, although there are a myriad of venoms, they have strong bias for systems in the prey that are essential for their living. So there is a large convergence of targets, although a large divergence in the chemical structure of their toxins. In the case of snakes are a little bit boring regarding to other concept in comparing with other venoms. So I would say it's a medium complexity proteomes formed by no more than 1 dozen or so of different toxin families, although their complexity relies in how they express, they combine different of these families into their venoms. Not all the families are in every venom, and also on the complexity, on the diversity of proteoforms that have evolved in every species in the venom gland. This is just a very recent paper on the evolutionary structure of three-finger toxins. So it's very interesting to see that evolved almost 340 million years ago and have been neo-functionalized in snakes since more than 50 million years. And this is a kind of tree of the family that probably is not even updated, but it's very interesting to read this paper. These toxins, they not only vary in their chemical structure, but also structure, and they associate in different manners, creating different epitopes that are involved in losing strength. Let's see. And different epitopes, and this means they have different opportunities to target different receptors, different binders, and so they diversify the toxic, the multiverse of functions that they possess. We also know something about the diversification mechanisms. They are genomic and post-genomic mechanisms, and here are some examples of gaining and losing genes in the genome, just stochastic in apes, stochastic variation, but also microRNAs have been shown to be involved in the few occasions or when they have been studied, and I think this would be also a big area of research in the future. Now that we are witnessing the emergence of genomics and comparative genomics in snakes, and this would be continue steadily rising in the future. So this is one of these conceptual slides just to remind us that we can divide toxinology in anthropocentric bias. This is what affects us, but mainly toxinology is also the study of toxins is also for understanding natural selection and prey, the way the prey are subjugated by the producers of the toxins, and how so in ecology in the ecological setting. So it's important that one divides the whole toxinology field in where you need to know the history of the preys and the interactors, and where you don't need to only what the toxins are doing to your body. So these are the two main areas that I wanted just to divide this. We're more in this talk, we are more biased against the anthropocentric biased toxinology. You will see that everything that we have developed can be applied for both, but we're applying in a translational way to human envenomings. These are the pillars that we will use in our research. We have here we have here a venomic platform that I will speak a little bit about it, and issues that still are there, not only conceptual, but also in the practical in different labs. We have toxicovenomic pillar that I will also speak about this part of our platform, and of course antivenomics, which is the translational venomic part of our platform to investigate and interrogate antivenoms about their efficacy and their backing in efficacy. So first, concerning venomics, let me say just that we have two different, in mass spectrometry, we have two different works, two different ways of interrogating the proteome. We can interrogate it from a bottom-up platform. This means you take the proteome, cut it in pieces, and then try to recompose the puzzle from the analyzing the pieces. And we have top-down, which is what I guess would be the future of the not only of genomics, but also of proteomics in general. That is, you just isolate the whole molecule, and everything that you do with these molecules will belong to the molecule. Everything that every piece that you characterize is from the molecule and not from another proteoform that could be in the solution. So you isolate the protein and start working from the intact protein and not from the intact proteome. So this is the workflow that we devised the years ago, and it's a low throughput workflow where you separate by reverse phase, then you separate the individual components of the reverse phase peaks, quantify this by area, quantify this by contribution, by densitometry and contribution of individual bands, and then you cut each of these bands. And if there is a transcriptomist, you will have a good database to interrogate. If not, we go de novo sequencing, and at the end, we piece up together the protein from the different families and draw these chart pies that just show the relative abundance of genome components in this particular proteome. So this is, and one thing that I wanted also to insist is in this mass profiling. Mass is the only feature of a protein that is absolutely specific for this particular protein. So there is no two proteins that have the same structural formula, which is a proxy of the sequence plus the chemical of the sequence, the length and the chemical structure of the sequence. And this is absolutely specific for each protein in a proteome. So if you identify here a mass and 10 years later, another guy in another lab identified the same mass, need not to characterize again. You can be sure that it is the same protein. So this is something that is important, and we rely also on this, and we work always when we can, we determine the mass. So this is just this. Let me just here are a couple of papers on the fine isotopic distribution and limits to resolution, and this review article that we wrote on watching a mass that I recommend you to read. And you have questions, just contact me. We can talk about this. But it's very important to be familiarized with mass when you are working in mass spectrometry. So this platform allows also for working on individual venoms in quantities. It could be at the level of your sete of the venom that you just. This is a bicephal snake that just lived a couple of days and then died. But in the time, we were able at the Instituto Clodomiro Picado to get some venom, and you see this is a comparison of the venom that was obtained from the left versus the right head at this small age of the poor guy, and measuring the masses, we could also in these peaks over here, we could also determine that it came from the Pacific Versant population of Bothrops asper, because these masses are specific for this particular population of Bothrops asper in Costa Rica, and also identify differences between the siblings and the mother. So it's quite, you can do a lot of things even just with the mass. Quantification is something that I wanted to insist here. We use the, as I said, the area. This is the first level of quantification, the percentage of the area. Then we divide this percentage by the percentage of the different proteins in the bands when they coalesce in the same peak. And then if two proteins or more occur in the same band, then we use for spectral counting or quantification by spectral counting or intensity of the ions, of the specific ions for each of the proteins. So we have these three levels of quantification. This is a very low throughput strategy, but works. So then, if you compare the low throughput with and the resolution that you get from bottom up with what you can get by working top down, where you can select here from the venom inside the mass spectrometer each of the proteoforms and just produce the degradation one by one, and then you can get this resolution that you will never obtain in a bottom-up approach. So for me, this is the future. Here is in comparison. You have by top-down, we identified, in this case, 268 proteoforms, but only 38 in the bottom-up approach. So it's much more resolutive. Let me also say that a trend that is now in the field is to take the whole venom, chop it with trypsin, inject in a single injection into the mass spec, and or with a column, separate peptides, and just go one by one and identify in a shotgun way, and then the problem is how to quantify this. So people, what they do is from this chromatogram, after the identification, from the signal, the MS1 signal or the MS2 signal. This is the fragmentation of each of the peaks, and these are the intensity of the intact components, the peptides that hit the detector. They produce an extracted ion chromatogram and quantify with algorithm the individual the quantity of each protein, which is also relative. But here the problem is that you have here in the formulas that you can use for quantification in this manner is always a relation between what you observe and related to what could be observed. So if you don't have a complete database, you are biased against what you observe. This means that you are just interrogating a small part of the protein for quantifying what you are observing. But there are many things that you have not identified if the database is not complete that will stay apart from the quantification. And the consequence of this, it is other algorithms that are using, but all of them rely on the relation between what is assigned to the total proteome. If it is the database is not complete, of course, you have problems. So look at this. This is the same this is the same proteome if it is analyzed from a model organism. This is an organism where the database is not an issue. You have the complete genome or the complete transcriptome. All the databases is there. So you can assign for all the detected peptides, you identify a hit. In this case, if this were a non-model organism, this means that you have, of course, the same venom peptide digest, but here you detect the same, but only identify a few of them, not all of them. When you chart the pie chart, when you draw the pie chart, here the composition you see is absolutely different. It's 48% here because these are the ones that are identified. And here you have identified everything. So this is a strong problem. People still continue to work in this manner because it's cheaper. You need only to have one digest. You throw you send to the service. The service will charge you only for one experiment and not for 50, etc., etc. But they are populating a lot of fake results in the literature. So this is something that I wanted to tell into this audience. This is also the same. Another example from real life. In this case, this is this bilineatus snake, the venom of bilineatus. This is when you work out the proteome by our snake venomics platform, or in a shotgun using ion intensity extracted ion chromatograms. And here is the same for another snake, but here we use the spectral counting. So you see, it's the same bias always. So take care with when you evaluate papers like that use this quantification incorrectly. So in mass spectrometry, all the advances in mass spectrometry always involve hybrid machines that have the potentiality, or they have the advanced potential of using different modes of analysis in the same system. In this case, you know the cutoff that is a triple quadruple, and different other, the cutoff orbitrap, and so on. So we thought that it would be also nice to include another complication to a system. This is experimental complication that is add a third possibility of analysis, which is absolute protein quantification. So absolute protein quantification, the only mass spectrometry workflow that can do this is isotopically coupled plasma mass spectrometry, ICP, which was you only quantify atoms. You don't quantify molecules. The only that you quantify, and very precisely and in absolute terms, is atoms. So in here, what we do is this part of the system, which runs in parallel of the molecular mass spectrometry. This is elemental mass spectrometry, and will only detect sulfur. But will detect sulfur in parallel to what you are identifying and quantifying by bottom up, and identifying by top down the mass of the proteoform. So here you identify the proteoform, then this means you know the sequence, and you can calculate the stoichiometry of sulfur from the cysteine content plus the methionine content. Here you are quantifying the sulfur. It's easy to correlate this with this and say, well, the absolute sulfur concentration is divided by the the number of sulfur atoms that you have in your molecule and taking into account the mass, and you can quantify in absolute terms each of the proteoforms that you are analyzing. So we have done this for several venoms, works fine, and we will continue to work on this. And the things that we are now trying to implement is based on extinction coefficient. So from a protein with its sequence has an extinction coefficient at the wavelength that you prefer usually is 280. And once you determine the extinction coefficient of a protein, you can use it once and again every time that you are using this protein. You don't need to measure, you don't need to measure Bragg4 or any other colorimetric reaction, which, by the way, for short proteins do not work the same as for the standard protein that you use to calibrate the response of the reaction. So you can calculate, and actually with this formula, this is 1%, 1 milligram per ml protein is n times the tryptophan residues in its sequence times 5,500 plus the number of tyrosines plus this figure plus the number of disulfide bonds plus multiplied by 125 divided by the mass in Daltons of the protein. And this gives you a figure that is very close to what you determined experimentally. This is the experimental determination of the spectrum of the extinction coefficient of this venom. And if you just take the extinction coefficient calculated for these 4 major components, and you know the percentage of abundance and you know their masses, you can calculate the extinction coefficient for each one and the contribution to the extinction coefficient of the venom, and you obtain 1.31 instead of 1.34, probably because we have not taken into account other components, minor components. And the same for this other here from Naya nigricolis, 116, 106. Also the same from these 5 components. So this means that it is feasible, and if it is feasible, perhaps we can also translate it to the same system where we can calculate, where we can quantify a proteome in absolute terms. But here, what we want is, instead of determining, where are you? Instead we will we want to combine these 3, the elementary and the molecular mass spectrometry systems, to calculate an extinction coefficient for the peptide bond, not for each of the proteins, as I saw in the previous slide, but for the peptide bond. The peptide bond is the same for every protein. So you have n minus 1 peptide bonds in the sequence. So if we can here work out the extinction coefficient for the peptide bond, then we can translate this from the absolute quantification to let's say in another manner. We can apply this to quantify continuously the trace of the chromatogram according to this extinction coefficient, but in this case would be 214 instead of 218. So we can convert the qualitative trace of a chromatogram into a quantified trace at every point. And this is this would be in advance because you will get absolute quantification continuously across the chromatogram. So this is something that we are working with with colleagues in Oviedo University, which are experts in ICP, MS, and we provide the venoms and also divide the work between each other. So let's go to another pillar of our. Let me see. This is okay. Of our venomics. This is now a translational venomics workflow that we need to have into consideration for what I will tell you, which is the major topic of the talk in the second half of the talk. So here is easy. Toxicovenomics. This is a word that my colleague Bruno Lomonte coined for the profiling of a venom in terms of the toxicity of each of the peaks. So you have a chromatograph, you have here a venom, you separate the peaks, you know the relative abundance from the chromatogram of the peaks, and you isolate the peaks and determine the toxicity of these peaks. In this case, is LD50 across the different components of a venom. It is clear that those components that are more abundant or low abundant but with a high toxicity will be more relevant in an envenomation setting, in envenomation accident than those that have low toxicity or low abundance and low toxicity. So you can divide the LD50 in this case in here in moles per gram of body weight, divided by this, divided by the relative abundance of the different toxins, and then you have a kind of toxicity score which tells you which are relevant, medically relevant for the human. Those that in this toxicity score is higher than 1 LD50 for human, not for a mouse but for human. So we will come back to how to convert mice data into human data. So having identified here let me see, this is also a slide where when you, where are you? Yeah, but I wanted to. Hey. Okay, let's stay with this one. So this is the LD50 for individual components. This is the percentage of each of the components. And now if you multiply, if you calculate here the contribution of this portion of the venom to the LD50 of the complete LD50, the LD50, the whole venom, which is 575 here, then you see these are the relative LD50s, the relative contribution to the LD50, and if you sum up all of them, you end up with a value that is almost the same as for this. But now here you have distributed in terms of each of the components. And this is the data that you need to evaluate which toxins you need to neutralize the action of the whole venom. I promise that I will talk about allometric translation from mouse to human. So this is something that is being is in the field since the 30s in the last century, and what people have found is that there is a linear relation between this one. Let me see. A linear relation between the body mass and the basal metabolic rate of the organism. You see here from unicellular to the elephant. And from this, it can be calculated from the weight and the square meters of the body surface area. You can calculate factors, kilograms divided by square meter of surface area. And for human, for adult is 37, for child is 25, for mice is 3. This means that you just need to apply this formula, the human effective dose in milligrams per kilogram equals the animal dose in the same units multiplied by the ratio between their kms. Let me just put an example. This is X ocellatus. Okay. This is. Pero quiero. Ay. You guys got it. So this is X ocellatus, which for the mouse has an LD50 of 1.4 micrograms per gram of body weight. This means 28 micrograms per mouse of 20 grams. Okay, if we translate this linearly to human, this 1.4 micrograms per gram multiplied by 60,000 grams of body weight of a human would be 84 milligrams, the LD50 for a human of 60 kilograms. 84 milligrams. These guys do not produce this quantity of venom. Here is the venom that different, in this case, X ocellatus, but other X produce, and this about 11 milligrams. So this means that if you translate directly, it is impossible that this guy which caused so much disease, so much envenomings in Africa would be of medical irrelevance. However, if you translate the LD50 by multiplying by the ratio of the kms, then it's 0.1135 milligrams venom per kilogram of human, which means 6.8 milligrams will correspond to 1 LD50 for human. Now we are seeing more sense in the data corresponding to the clinical setting. So this works, and we use this always for translating the data from. So, toxin resolves antivenomics. Antivenomics is very simple method where we immobilize a ligand, in this case the antibody that we want to test, and incubate different columns with different amounts of venoms. Then quantify the relative abundance of what is in the flow through of the immobilized antivenom column, the affinity matrix, and what you can obtain after washing the column, eluting the column. So, and then you have here what is bound, and you have here what is not immuno-retained in the column. And with this data, you can plot here for the different amount of venoms incubated, what has been bound, and then determine what's the binding, the total binding capacity for this venom of this immobilized antivenom, which is the sum of the different components at saturation in the affinity matrix. If you don't saturate completely the matrix for this particular component, you can extrapolate to infinite and also can retrieve this figure. In this case, by just adjusting the saturation curve. And then you can calculate, see if you know the amount of milligrams that you have in a vial and what is the concentration of the vial, and with the total amount of protein that you calculate from here, you can calculate what's the neutralization capacity per vial in terms of milligram of total protein per gram, or whatever other unit you want to use. We use gram because it's more comparable between different products. If you do it by microliters or by milliliters, then if you don't normalize by the concentration of each of the products, which in many cases is not known, and if you don't determine it, then it's a mess. You can not compare pears with apples and so on. So we always do comparisons in terms of milligram of protein per gram of antivenom, and then you just multiply by the concentration in the vial and have the milligrams per vial. So the beauty of this antivenomics analysis is that it tells not only what the antivenom is able to bind and the total potency against the venom, but also against each of the components. And in this case, you see here that this peak is not retained, the 2.4 milligram incubation is not retained at all, and here the same happens. So you can increase the amount, it will not happen. This is simply a protein that has not binding antibodies in the antivenom. So it tells you something that needs to be improved for this particular antivenom. So you can do this for every antivenom. So now we know the three pillars that we'll be using in the following slides, and how we combine one with the other to analyze the efficacy of antivenoms. This is, we have seen several times. I don't need to waste time. This is to change from topic now to the second part. We are now analyzing antivenoms, venom versus toxins, or vice versa. So what's in a vial? Well, this is the sad situation that we find after analyzing all the commercial antivenoms in Africa for in the pre-qualification WHO program. And the sad situation is that almost no one can be recommended for treating elapids, and for viperids, not all of them, but most of them, but not all of them are effective. So there is a huge problem. Well, at least for the whole venom or the whole bite that the snake can produce. If you are lucky and the snake just gives you a defensive bite or something like this, and your symptoms are not bad, of course you can use the antivenoms, but in high amounts. So then we started to think in a proof of concept that is maybe that it is not only the antivenom the cause of this disaster. We believe that the antivenoms contain neutralizing antibodies, but not in the sufficient amount. So here the proof of concept was that current antivenoms can be refurbished to clinically effective products. So without any other technologies, just taking outdated antivenoms and try to produce a useful product theoretically, because we will not be doing clinical trials at this stage. And this is the team here. We have Jordi, Davinia, Jania, Carol, Livia, that is now retired, and me. So we started with a compositional homology and the toxicogenomic profile of each of the species in this phylogenetic tree, all the species in Naya. Some of them we could not get venom, but for most we get venom, and also include in the study all the Dendroaspis species or subspecies. So here we determine all the masses, we interrogated transcriptomes, we identify the toxins, and the same for the, we determine LD50s in many cases for individual components, relative abundances, and so on. Then, with this data, we used, we decide which toxins we want to isolate and to work in the next step. So here we were conducting a large-scale purification of all the medical relevant toxins that the previous analysis provide us information. An important thing here to consider is also how can you go beyond stoichiometry. For a Fab2 product, 1 gram of FAB2 prime, 110 kilodaltons, can bind 18.2 times 10 to the minus 6 moles of antigen. This is the number of antigen binding sites that it contains. You cannot go farther than this. You cannot bind more. So this means that if you want to target a three-finger toxin, a gram of this FAB2 can bind 123.8 milligrams, no more than this, stoichiometrically, with both sites occupied, and the same, and for PLA2 is 247, and for the dendrotoxin is 111.6, because this depends on the mass. However, if you select properly the toxins, then you can increase the binding capacity in terms of venoms. The other was in terms of toxins. But if you want to increase the not the stoichiometry, but the binding towards a whole venom, if you select here, let's say that this toxin, this alpha neurotoxin, which is 15% of the total venom, has the major, all the sole toxicity of the venom. In this case, the 123.8 milligrams that you bind here correspond to 825.3 milligrams of whole venom. So you increase the binding, apparently the binding capacity. And the same is here. In this case, if all the toxicity is in this molecule, this means that with this 32% abundance, 1 gram of anti-PLA2 antibody until this one will have a maximum binding capacity for this venom of 770 milligrams. So you need to have this into account to select which antibodies, because you want to have the less amount of antibodies with the highest binding capacity for what you want. So this is important also to know. Here is a general formula. So then another thing that one needs to have clear is how to maximize the smallest amount, smallest set of antibodies towards the highest number of toxins, of relevant toxins. And the only way is paraspecificity. You need to identify the highest paraspecificity. This means the minimal number of toxins that you need to isolate for purifying the antibodies that are paraspecific for a broad and the highest number of toxins. So this is also something that we took into consideration. So with this, we generated toxin class affinity columns for the 4 different class of toxins from Dendroaspis and from Naya, with this minimal set of toxins immobilized in the column, in the affinity matrix. So we use outdated antibodies and serial purification of antibodies from these outdated commercial antivenoms. As a whole, the mean purification antitoxin antibodies was between 3.9 and 12.9 of the total antivenom, depending the antivenom that we had for the purification. So then we determine the neutralization activity of each of these 4 antivenoms, either individually or in combinations, and the results are here. You see all the different venoms from here were neutralized. Here with this only very poorly, but when you combine with others is effective, and so on. So the proof of concept needed only to test formulation, and the formulation and for the formulation we had this 55 milligrams per ml vial with the formulation that we gathered from the previous slide, and with this, these are the milligrams per gram of venom that were neutralized from each of these species, and this is the potency. Potency is not the same as CD50. So if you use N LD50s, the potency is the LD50 divided by N minus 1. So if you use 5 LD50, the potency, which is what neutralize 100% of the venom, not 50% only, would save the whole population of 50%, is the ED50 divided by 1.25. And if you use 3 LD50, would be divided by 1.5. So then we here also use 5 LD50s for rescue and for this formulation for rescuing the toxicity of 5 LD50 of this one. So with this, we could rescue all the mice except one stupid mouse from the Naja arabica, injected with Naja arabica venom. So, but outliers you find always. But in principle, I think we were happy with this proof of concept. Another thing that we did during this stage of the project, I still have 10 minutes or so, in this stage is to determine the amount of venom that species can inject theoretically. To do this, what we did is just bother the snake and let it bite, and then determine what the snake has bitten into this jar. And the same, if you look here, here down, the resolution here is not, but you see the droplets come down. So these are the venom yields that we obtained by combining the experiments that we did with the data that we gathered from the literature, which we had to do a kind of archaeovenomic by bibliographic search, and finding we found incredible amount of data, but some of them in journals that are not available in any library. So in many cases, I went just and bought a book because I was only on a book, or search libraries that could have this. So, okay. So we did this, and then we have better figure of the total amount that we need to neutralize in the worst case. here is a complicated table, but is simple to explain. Here we have the potency of the formulation that I just showed you, and calculated here the grams of antivenom that would be needed to neutralize 100% of the mean venom yield. In most cases, it's below 1 gram, except in these cases where the calculation, because they have a lot of venom or because the neutralization potency is not as high as for others, we would need, in this case, 4.8 grams for the venom yield, 1.2, 2.4, etc. But it's a reasonable numbers in terms of gram compared with what is in the market, even for the good antivenoms. So just briefly, what we have been doing from May 23 to November 24, the second part of this project that was generously not as welcome trust, but generously financed by the Global Snakebite Initiative. And so here what we did is calculate from the previous results, try to construct a single column that will contain not toxins, but the venoms that contain the amount of toxin that we would like to have immobilized to isolate the formulation of antibody that we got. So emulate, but in a single column, and immobilizing venom, which is much easier to immobilize than isolated tons of toxins. So for this, of course, we calculated the percentage of abundant and so on. So here, at the end, we mobilized these equivalent milligrams in toxin of short neurotoxins, long cytotoxins, PLA2, the dendrotoxin, 601 milligrams of in total in the single column, which had a 14.7 plus minus 2 milligrams of venoms per ml. This was the saturation of antivenom binding of these antivenoms that we received here in, I think that here were about 60 liters of hyperimmune plasma that we process in the lab with caprylic acid and purify the IgGs and used for isolating the binding, toxin binding activity. So from Silanes and also from premium serum. So then we went to, with the product that we obtained, we prepared it and went to interrogate what we had extracted for antivenomics. So to have an idea of how well or how bad this pool of toxin antivenoms that we isolated from the single column bind to the different venoms. And here are the results. The results are preliminary, of course. Here is the first formulation, but this formulation gave good results for short and long neurotoxins. This means that for me, good result is that the maximal amount of milligrams of toxins bound per gram of antivenom lies in the middle of or in the range of the milligrams of toxins per venom yield that need to be covered. So here, for this, in almost all cases, is in the middle of the range. The same here, and for the different toxins, the alpha and cytotoxins and so on. Here you see that the situation is a little bit different for many cytotoxins from NAJAs. That was suboptimal, the binding. For some was okay, for some others were not. And particularly for the one over here, these cytotoxins. And for the dendrotoxins, for polylepis was okay, and for the other was also not as bad, but also suboptimal. So if we observe the antivenomic analysis, it is clear that all this part here of the chromatogram is well recognized, and the major drawback in recognizing in binding is here. This is 29% of the venom, but is only recognized in a few percentage. So, and this means that you will have not retained 51 to 66 in the bite, and this will cause problems. Okay, no, here. If we observe how is in the other venoms, in all of them is the same. 22% bound, 35% bound, 24, 34, and so on. So this means that we identify with this antivenomic analysis which toxin need to be improved in its binding to the antivenom, and are for most of the venoms the same toxin. And it's a toxin that you can isolate in hundreds of milligrams quite easily with a preparative reverse phase column. And the same happened here with the polylepis. This part, which is present in the non-polylepis species, in the others, is the one that is less well recognized than the other. And you see here the reason. This is well recognized. These that are all very similar between themselves and were not included in the affinity column are less well recognized, need to be included in the affinity column. So in parallel, and just to finish the talk, we are also collaborating with producers, with two producers, to collect the peripheral lymphocytes from hyperimmune blood of horses, and isolate the clones and try to do the same as the affinity column, but in cell culture. So in principle, if you put all the clones that you need in the culture and you know the expression of each of the clones, you can put in a way that at the end you get here the antivenom ready for purification without the need to go this for each of the components, etc. And also the toxins that were less well recognized were also purified for either giving to horses as supplements or immunize horses only with these toxins. So we have agreements to do this. These are the PBMCs here that were isolated from the horses, and here they were prepared for shipping, and 1 week before coming here, they arrive in the lab, and we gave to our collaborators that will produce the cDNA libraries, etc., and tell us how good or crappy these mononuclear cells are. So with this I think I have finished. I just want to say that this lecture is dedicated to how is the name? To Royan Taylor, who was the director of the BioChem snake farm, which are collaborating with us now, and died suddenly a couple of years ago after a couple of weeks later of the presentation of the work of the hoja de ruta of the guidelines of the WHO for the snakebite program of WHO. So, and with this, we are 5, but along the life of the lab, we have many collaborators, and here are some. Also many from Brazil, and just also to say thanks to everyone that contributed over the years to what the lab is now. And here, people from other states that I don't have. Thank you.
Thank you, Professor Calvetti, for the great talk. I will open to questions now. Paulo, por favor.
I was watching your list of Naja, different kind groups of species of Naja in Africa. You didn't put Naja Coachia from the India and the China, the Asia. Why not? Because your antivenom could be for because your project for Africa, but you could have a big population over there.
Sure.
Why not? There is some similarity between Naja Coachia and the other African and Asian.
And for sure, there is some paraspecificity. For sure, I don't know how much, and if it is the same or if it is enough for the same formulation being used in Southeast Asia. But the concept here was first Africa, and then when it is ready, we will test against the paraspecificity of other species and determine where it can also be used, or eventually produce another product for another. But this should be produced with the producers. So we will not do an antivenom in our institute or in other facilities in Spain. So what we want to do is to be a kind of investigation and development department for the producers, so that they can we can transfer the technology, we can transfer the methodology, we can help them in implementing this for they to produce the antivenoms. I will not put a cent here in my pocket. I have enough with my salary and with my retirement when I retire.
The very good similarity. China has a good economic, and also India, but they key producers.
And the Arab countries also.
Yeah.
In some of them you can live happily, in some of the other not.
Thank you very much.
You're very welcome.
Thank you, great talk. I would like to ask, how do you think whether it not increase enormously the price of the product? Because no.
No, because everything is okay. Everything. If you use the affinity column, the cost is the cost of producing the column.
So it is not that much.
A column. We have used these columns for 30 something times, and with the same, without losing the affinity and the yield for recovering the antivenoms. So this means that you can produce solid and columns, and once you have a column, you can use for many times. So this is not a problem. And we are working in parallel also, not to implement something that cannot be implemented in the producer. First, give a better solution, and then try to give another alternative, or even two solutions, or even a better one with the cell cultures and so on. But
and another one question concerning
limited.
the identification of toxins with the proteomic bounds, whether you think that it could be done also with Raman spectroscopy, because I've noticed very sporadic publications concerning
But
this
Raman
vibrational spectroscopy of the protein
I don't
bonds.
know from any law that can identify a protein by Raman. I don't know which kind of figure would you use, which kind of parameters would you use
to
identify the
with tandem, as you've shown, when you use the normal proteomics identification with
you mean for
UVVs.
quantification?
Yes.
I have no idea about the properties of Raman or how to implement here, how easy or not would be. I know that the elementary mass spectrometry is well established in the field, in organic chemistry, and works, and you can rely on the standards that you have on the market for calibrating. You can rely on now from the periodic table with a triple quadruple that eliminates the interference of adducts that can have the isobaric masses with the atom that you are trying to investigate. Has solved, so you can determine any atom in the periodic table except carbon, hydrogen, oxygen, and nitrogen, which are in the ambient, and you cannot retire this from the flame of the instrument. So you are simply cannot have all 99% contamination with these atoms. So, but the others, you can determine all of them quantitatively.
Thank you very much.
You're welcome.
Any other questions?
Okay. Alexandre.
Good morning, Doctor. Thank you very much for your speech. Just one question around what the doctor from Armenia said about the affinity columns. What about the compounds that are strong, like react with the antivenom column? I mean, metalloproteinases. You in your articles talk about there are some compounds that have like strong affinity with that antibodies, so you have to do like basic washes. I don't know, strong washes with basic compounds, and it will like remove in sometimes the antibodies from the column.
No.
And it reduce maybe the sensibility or the affinity.
No,
the
the antibody, you will not remove it from the column because it's covalently bound. And it's stable covalently bound in the column, as far as we know from all the dozens of runs that we have done one after the other. We take aliquots from the eluate and never found a single antibody molecule. The rest, it is true that in some very high affinity binders can bind, are the minority of them, so are not of great concern. And on the other hand, what you can use in the industry is done is to wash the column after 2 or 3 times, wash the column with 0.1 normal NaOH, and then equilibrate again, and then you are at the same level of binding that before. So this is something that you need. Even in reverse phase, we do every three runs, we put a washing run to release everything that was unspecifically bound to the column. Yes, this is very common.
Okay, yes, because it is a problem that I have, for example, in cases of Acrocorda venoms with that compound that is strong, like binding with the column. But for example, if we don't wash the column, I mean, the affinity, for example, the compounds that are hardly binding to the column will be in the part that is not like the no binding part. If the old antibodies are busier with other molecules that we can elucidate. So you recommend always do that washes,
The worst I recommend always doing. Individual cases need individual solutions also. But on top of this.
Thank you very much.
Okay, unfortunately, we don't have time for more questions, but you can talk to Professor Calvetti later.
With a cachaça.
Thank you very much, Professor Calvetti. Great talk.